All right, so the CNA tutorial. If you wanted to run this on your personal laptop, there's a worksheet available here you can access. And this is the workflow that we'll be going over. CNA stands for copy number alterations. So there's about 7 steps to this, starting off with pre-processing, collecting our read counts, creating a panel of normal, and then denoising our read counts. There's also a second portion where we're merging our collected allele counts into our denoise copy ratios. Going down here, our first section just talks about the differences between the workflow in GATK4 released version versus the GATK4 beta. And mostly of these are the name changes. So there's a name for the GATK4 release along with the beta versions, and then a description of what these tools do. All right. And here's a little bit of information about the data sets that we'll be using in the tutorial. We are working with whole exome samples, and we're going to be switching between a subset data along with real full data. And the subset data is just to get through this tutorial a bit faster. The panel of normals that we'll be creating for this tutorial is coming from phase 3 1000 whole genome project aligned to GRC38, along with interval files, which is a mixture of the HCC capture kits along with 1000 whole genome exome targets. All right, so we need to go ahead and get things set up now. The first thing you need to do is make the sandbox directory. You probably already created it in the previous workflow, but shouldn't. Yeah, you still need to create the sandbox directory or this command, because we will be creating a CNA plots directory within our sandbox directory where we'll store the plots that we generate. So go ahead and run that command. Awesome. Okay, after that you should get CNA plots. Don't worry about this kind of warning. No need to worry about it. Next, we're checking whether or not we have access to our Google buckets, and you should, after entering this in, you should be able to see the directories inside this GATK tutorials Google bucket. So nothing being done here. All right, so there are some R dependencies that some of the tools that we use in this tutorial needs, and we're just installing these with this command. And these are for plotting our data. Awesome. I think for the most part, everyone should have their packages installed now. I'm going to move on to downloading data. So some of the commands in this tutorial are able to access files inside a Google bucket directly. Others do not have that update yet. So for those commands, we'll need to download the input files to our local machine, or our local notebook machine. And that's what we're doing here. Making a directory and then using gsutil to copy CNA inputs into our a directory. Awesome. So that was all the setup kind of commands that we've done. We're going to move on to the actual first command for our workflow. Before I move on, is anyone having trouble with the setup commands? No, great. All right, so here is again, our workflow. We are running pre-processing intervals where we are adding padding to our intervals. To run this command, you'll use this tool, preprocessing intervals. We're providing it the interval list that we want to have padding on, along with a reference file. In this case, is HD38. And we're indicating how much padding we want using this parameter padding. Since we're using exomes in this tutorial, we'll add in 250 and set the bin length to 0. There's another parameter here called interval merging rule, and it's set to overlapping. So by default, this tool will merge any intervals that are right next to each other. By setting this to overlapping only, the tool will only merge intervals that are overlapping one another. And lastly, the output file is the other parameter. So go ahead and run this command.
I was wondering, do you know about the interval overlap setting? If there's like a recommendation on how much overlap is meaningful, or is it like a portion of the interval size?
I wouldn't know over the top of my head. We set this in, overlapping interval list or parameter just to make sure if we have any overlapping intervals, they are, merged. If they're adjacent to one another, they're not merged.
So is there like a minimum amount of overlap before they're merged, like 2 base pairs? Or?
I'm pretty sure if there are intervals that are overlapping, they're merged.
Okay, just merged. Okay.
Yeah, and so I'm familiar with this kind of tutorial. If you have really deep burning questions, we do have that channel, GATK channel, where the actual developer of the workflow said he'll drop in and answer any of your questions. Is everyone set for the next command? Yeah, looks like it. All right, and before we move on, let's try to answer this question. There's a few questions in this tutorial. We'll try to get through them, but we might not due to the interest of time. But for the first question, it says, peruse both of your intervals, both before and after you've executed them, and check whether or not you have the same number of intervals. So in order to do that, I will create 2 cells using this insert button. So click on that. And then I'll add in an exclamation point to indicate that I want to run a shell command, and type in gsutil cat, and then the path to the input file, which is listed in my command up here. Now I'll paste that there. Then I will pipe that through grep to look for any @ signs, and then pipe that into word count to count any lines. And I'll press enter. No file directory. I did, and then I got logged out. Ah, there it is. You're right. Awesome. So there are 11,000 intervals in the original interval list. How about the output? So instead of using gsutil, I'm just going to enter in cat because the file is saved in my sandbox, which is on my notebook machine. And the path to that file is listed here. And do the same thing. Grep plus word count -l. Awesome. So looks like the intervals are the same number of intervals. Any more questions? Here's a quick kind of pop quiz. what does the IMR overlapping parameter do? Questions from that? No? Anyone? It's the, say that, what? Right, it was similar to what I was saying earlier, this interval merging rule. It prevents any adjacent intervals that are right next to each other from overlapping. It only overlaps ones that are overlapping. Right, moving on. So we ran preprocessing, the preprocessing tool. Next, we want to collect our read counts from our tumor and normal samples. To do that, we use this tool called collect read counts. We're going to run this twice, once for tumor and the other for normal, and providing the same similar parameters. So input is our tumor BAM. Our interval list is the padded interval list that we just generated. Next, we'll have the reference FASTA file. And we have a parameter called format, and it's labeled TSV. Normally, the output would be written in HD5, HDF5, which performs better. But for the sake of this tutorial and for users who might want to look inside the file, we'll leave it at TSV. And again, we have the overlapping only parameter added. And then we have our outputs file. So I'll run both of these. Okay, so once you've executed those two commands, you'll have a normal and a tumor TSV counts. And if you were to graph these or plot these files, you'd get something like this. You've seen this in the presentation previously. These are like the x-axis would be the chromosomes, while the y-axis are the counts at those interval lists. And you really can't really make heads or tails of this when you're comparing it from the normal and the tumor. So we'll be denoising these plots to make it more legible. To do that, we're going to use a panel of normal. So here's the workflow again. We've collected our read counts, and we want to denoise our read counts. But before we do that, we'll need to create a panel of normal. And when we run this create panel of normal tool, we'll need to have a list of normal BAMs that we want to include in our panel of normal. And after that, we'll need to collect the read counts for each of those normals. Once we've collected the read counts for each of those normals, then we pass those into our read count, the create read count panel of normals tool. Where is this? Okay, so create read counts. Due to the sake of time, we won't be running the create panel of normal. We do have the command in order to do so. So here is the command, GATK, and then create read panel of normals. Once you've run all the normals through the create or collect read counts, you'll list each of those files under here. So I indicating your input file, and you'll list the inputs, file 1, clean, HD5 read count, and then all the way up to file 40 read count. Okay, and you can, there's also this parameter called minimum interval median list, which allows you to, just reading off this tutorial here, the argument for it is 10. If you decrease the value to 5, it will retain more data because you're essentially saying for each of the intervals, if I see a median that's kind of low, it's okay to include it even if it's around 7 or 6. So you're just decreasing the minimum interval median list, and it's normally used for well-curated cohorts. We do have an example of running it in the tutorial, running this create panel of normal in the tutorial. Again, we're not running 40 samples here. We're just using the normal counts that we created in our past example. So this would be the normal counts TSV. If you just run it, it will produce an output called normal dot, and it will create a panel of normal with one single sample in its panel of normal. Awesome. So that didn't take too long. Okay, and here is the same command again, create read count panel of normal, using real data this time instead of the subsets data we used earlier. And we're adding the minimum interval median percentile parameter here. Okay. So there's a question on whether or not it would be okay to, which one would work best, 40 sample panel of normal or a matched normal, and why?
Shouldn't we be doing both?
Yes, correct. That's actually, yeah, the suggestion is to try to use both. Never use just the matched normal. It doesn't have that much information, or it doesn't work well when denoising your parameter or your copy ratio. So it's best to use both a 40 sample panel of normal along with your matched normal.
Great. I kind of asked this question again, but let me reformulate. Is it better to use 40 independent normal samples, or can we if we have, let's say, 40 tumors, can we just reuse the 40 germ lines?
As in individual, what do you mean by individual?
So let's say we have 40 patients.
Yeah.
We're sequencing their 40 tumors. And then we have 40 germ lines, like 40 blood samples associated with them. Is it better to use the same 40 blood samples for our panel of normals, along with matching these ones individually with their tumors? Or should we preferably use 40 independent ones that are also run through the same pipeline, but not the same patients? Or are we losing information there?
This is a question I think you answered, or you.
Yeah, but before I just asked if it could be, I'm asking what's better.
I wouldn't know, like, can't say like for certain because I don't normally run these commands. Steve might have a suggestion.
I don't know the exact answer to that, but maybe Sam can answer.
Yeah, Sam Lee. So go ahead and put your question in that channel. Okay. So everyone should have already completed these two commands. We're going to move on to the next section, which is removing noise from our read count. So going back to our workflow image again, we've completed collect read counts, and we've also completed the creation of read count panel of normals. We're going to use our panel of normals in our this tool called denoise read counts to denoise some of the information from our read counts. So here are our two commands here. We have one for tumor and another one for our normal. The input that we're providing it is the read counts HD5 that we would have created from collect read counts, along with our panel of normals. And it's going to produce two files, a standardized copy ratio and a denoise copy ratio. So go ahead and execute both. Okay. Yeah, so it looks like it's a quick run. So everyone should have completed that command already. And there was a question about the number of eigen samples parameter. I was hoping it would be listed here, but it doesn't look like it is. If you do have a question about the default for any of these parameters, you could visit the GATK tool documentation site. And for this tool, or for any of our tools, there's the default listed along right next to the parameter. Okay, so we're at this point. We just finished our denoising our read counts. Next, we would go to model segments. But before we do that, let's go ahead and plot what we got from denoise read counts. And again, remember, previously we were graphing counts, but now we've shifted over to copy ratios. Okay. Right after, so once you've entered that command, you can view your plots by clicking these links at the right under your command. That up. So the plots that we generate are two, one for tumor and one for normal. And for each set, we have a standardized copy ratio and a denoised copy ratio. And for like pretty obvious, the differences between our normal and tumor is pretty, it's spiky for our tumor. It's pretty towards the center at 1 for our normal. And just to note, you probably already know this, but the standardized copy ratio is in blue, while the denoise copy ratio is listed in green. Great. Once we've completed the plotting, our next step is to run model segments to group our ratios that are similar to one another. So to do that, you will use these two commands. We'll do it one for our tumor and one for our normal. And I may not have said this, but probably you already know, the parameters that were listed here is the denoise copy ratio, which is the file we created from our previous command, the denoising tool, and just providing the output file along with the output prefix. Cool. So your command should have finished model segments. Next thing we're going to do is plot our model segment graph using this tool, plot model segments. We're going to provide it our denoise copy ratio and the segments file that was produced from model segments. And we have sequence dictionary from our reference, along with minimum config length. Minimum config length just helps to eliminate any mitochondrial sequences or unlocalized configs. Next, we will just provide the outputs, output directory along with the output prefix. Once executed, you'll have access to the plots that was generated. So looking at the plots it's generated, notice it's much more colorful and there's lines here. Like Steve mentioned, the color coordination just indicates the changes between copy ratios. And these are just the segments for each of the grouped copy ratios. Great. Now, if you are interested in having your copy ratios called for you, we do have a tool called Call Copy Ratio Segments. Fair warning, it's in active development, but you still have access to run it. And now what that will do is just add in the plus and minus or zero signs, depending on what whether or not it's an amplification or deletion or nothing's happening. All right, yeah, it's a pretty quick tool. All right, I'm going to go back to this file now, our image. We've processed our interval lists, collected our read counts, denoised it using our panel of normal, and created model segments, which we've graphed. In case you wanted to include your allelic counts, we'll perform this now in this section. So before we can include our allelic information, we actually need to collect it. And we do that with collect allele counts. We'll do that for both our normal and our tumor. The parameters are pretty self-explanatory at this point. L standing for the padded interval list that we have. I, our input BAM or our normal BAM, the reference file we want to use, and the output directory. And as a reminder, I believe these allelic portion helps to identify minor allele fractions, and also copy neutral loss of heterozygosity. looks like the, yeah, this command might take a while. While the normal portion is running, can anyone answer this one? For sites lacking alt allele counts, what is in the field for alt nucleotide? Here's a hint. Go ahead and try looking inside the file that we generated for our tumor. Here.
Where did we access the file?
Right. I'll help you out. So I'm going to create a cell here, and I'm going to enter in exclamation point to indicate I want to look what's inside the whatever notebook machine, cat, and then this is our output file listed here. So, for instance, oh, yeah, just realized. So once the second step is over, then you can look inside this file. Awesome. Okay, where did it go? Okay, so you should see something like this. So let's go down to our intervals, actual data. Okay, so again, the question was, for sites lacking alternate allele counts, what is in the field for alt nucleotide? Okay, so we'll look over here. If we have this, we have the naming for each of our columns. So we have config, position, our reference count, allele count, the reference nucleotide, and the alt nucleotide. Question was asking if the alt count was 0, what would our alt nucleotide be? The answer is N. It's right there. So yeah, anywhere there isn't an allele count, you'll get N for alt nucleotide. All right. So where are we? We just finished collecting our allele counts. Next, we're going to merge our denoised copy ratios along with our allele counts using model segments. And you do that with model segments. We're going to, as input, we'll provide the denoised copy ratios, TSV file, along with the allele counts that we just generated. We're going to do that for both the tumor and the normal allele counts. Next, we're provide an outputs directory, the sandbox directory, and the output prefix. In this case, it would be HC1143TN.
So she had a question here. Go ahead.
Yeah, I was just curious about the generation of the PON because it seems like a really important step to denoise the data. And so I was just wondering, like, how do you know, like, how many samples need to be put in there? And also, like, how can you evaluate, like, where those samples should come from? Is it just kind of a lack of choice in the available data sets, or?
The recommendation is 40 samples, and how you get it is you want to obtain samples that have the same, I guess, sequencing process as your case samples. So making sure the sequencing is right, and I think the other kind of important thing is to make sure it's not gender specific. You want to make sure it's both male and female. Anything to add, Steve?
Oh, that 40 number is for exome. So if it's genome, it can go a bit lower. That's
40 for exomes and lower for genomes,
yeah, a
correct?
bit lower. But usually, the more the better. Just costs a little bit more.
So it's basically a sample of variation from other sources, so
Right.
that your model can take into account that variation and realize that it's not relevant to the experimental questionnaires.
Right. Right. That denoise step using the control is making that assumption that the biggest variances from the raw read count
Yeah.
is from the systematic bias.
Okay. And that's why you're discounting much of the first dimensions of the PCA analysis,
Right,
because
right.
a lot of the variance is not coming from something that is a relevant factor in your
And
analysis.
there's a parameter for tuning that how many principal help and got the exact name of that, but it's on the slide.
Okay, awesome, thank you.
All right, so it looks like this model segment's command completed. I'm going to move on to the next part, which is plotting our data. So at this point, what did we do? We've run this model segments using our collector allele counts along with the denoised ratio, and we've got a combined version. Next, we'll use plot model segments to plot the merged versions. Do that by executing this command. Again, we're providing the denoised copy ratios, allelic counts that we got from our model segments, along with the segments information that we got from model segments, and then the sequence dictionary, minimum config length, and output stuff, the output directory, along with the output prefix. It looks like it completed. So I'm going to go ahead and open that plot. And this is our combined version. So we have our denoise copy ratio at the top and our allelic, alternate allelic information at the bottom. Typically, the 0.5 is indicating heterozygosity. Anything between 0.5 and 1, or 0.5 and 0, like around this areas, are indications of copy number evidence or incidences. Okay. This is the last section. Congrats, everyone. So,
Next, you guys, you can make it.
yeah, bear with me. So we have two more commands, and this one is performing allelic data, or using allelic data alone. We generally don't suggest this, but in cases where you can't obtain a panel of normal and you can't get that denoised copy ratio, you can simply run this bottom portion with collecting little counts and then modeling them as segments alone. So to do that, we're just going to go ahead and execute these commands. First, model segments. And for this model segments, we are giving it the allele counts, TSV, along with the normal allele count, and then the outputs directories. Awesome. So our command just finished, model segments. Next thing we want to do is plot our data using plot model segments. Let's start that up. And we're providing the allelic counts along with the segments that we just produced, and then the sequence dictionary, minimum consig length, and output information. So looks like it completed. We're just going to have a look at what's inside. And this is our plot with just the allelic data. And notice it's a much cleaner look than the combined when combined with copy denoise copy ratios. And that's because you're losing a lot of information provided by the denoised copy ratios. So we wouldn't suggest this typically, but again, if you can't obtain pan of normals to denoise your copy ratios, then it's possible to run with just the allelic counts alone. And that ends our CNA tutorial. Question?
Just in that last part, is that visualizing both the alternate allel and the non-alternate allel at each segment as well? So like, for example, the first segment there, that's 0.35, that's the alternate allel fraction.
Are you comparing this line to this line?
Yeah, what does the higher up line do?
It's, from what I remember, it's a mirror image of this. This is supposed to represent a minor allele fraction, and the middle is supposed to be just a regular heterozygous indication. But it's kind of just a double check, or it's a mirror of what's there at the bottom.
So these two lines, if you look at it, they will always.
So if you look at those two lines, they will always add up to 1, because the minor allele is a numerical definition. It's minor, right? But if you consider several SNPs, let's say you have, again, human, you have mother side, father side. On this side, you have mother is the the alt. And on this side, mother is the ref. We're talking 2 het sites. And if suppose the mother side chromosome is actually amplified, you can say on site 1, the father SNP will become minor. On site 2, the mother side will become minor. And so if we just only plot the technical definition defined minor, you would see it jumps around, right? But if we say only the alt allele, the minor allele and the non-ref allele, then we always have this. So we don't have this kind of like a jumping around behavior.